I'm very pleased to say there is more and more research being done on Duchenne muscular dystrophy, but how can you tell what's trustworthy and what's not? To talk us through all of that, Professor Annemieke Aartsma Rus, who's Professor of Translational Genetics at Leiden University Medical Center.
Thank you very much for the kind introduction, and also thank you to Duchenne UK for inviting me again, but also this year providing me with logoed origami. So there will be an expanding origami zoo appearing where I sit. Feel free to take whatever you like. Okay, Jared is going to take everything, but there will be more. So today I will talk about how to identify scientifically sound research. And as you know, research takes time. So research starts with studying the disease. We might have an idea of a therapy. We study this in cell and animal models. And if we test it in cell models, we say in vitro, in glass, in vivo, in animal models. And then we do the next research, as Michaela already said, clinical trials is research in humans. And all of this takes a lot of time. So then we say, well, what's in the preclinical pipeline? Please talk us through it. What is important? And that's really daunting because I just did a check. So 2025, Duchenne and therapy, if you go into the PubMed database, 529 papers. Duchenne preclinical, 71. Duchenne and mouse, 213. And Duchenne and trial, 174. So as a parent, how do you know which clinical trials are based on good preclinical research? And from the preclinical research, how do you identify which are the ones that you have to get really excited about and which are the ones that, well, maybe you just ignore? So that's challenging. So I'm a preclinical researcher, so I will mainly focus on preclinical research. But when seeing, well, if you have the option of being in multiple clinical trials, how do you choose the one that's best fit for you? Well, it has to do with the burden, etc. But there's also certain things that you can check. So you can check whether the company went to the TreatMD Advisory Committee for Therapeutic, TACT. And Volker Straub will present later. He is one of the initiators of TACT. TACT is a multidisciplinary group of people, preclinical experts, clinical experts, outcome measure experts, patient representative, regulatory expert, and they look at the development of a drug developer, can be industry, can be academics, and they say, how can we make this better? Is everything done? Did you consider everything? What are the gaps? How can you make your development plan better? And then there's the CAP, the Community Advisory Board, which is a group of patients from around the world, where companies can ask, so this is our plan, do you agree? Is this approach something that you would be interested in? And these are things that companies can do, and I'm not saying that if they didn't do it, the trial will automatically not be good, but I think as a field we deserve that companies and drug developers put in all the effort and use these tools that we have as a field to make the drug development approach better. So now focusing on the preclinical part. By popular demand, it's going to be interactive again. So if you were here last year, you know that you have to use your mobile to scan the QR code and to ask or answer questions. It's anonymous, and some examples are extremified to really make a point. And sometimes you may get the answer wrong in your idea, but you will probably learn a lot more that way. So don't worry about that. It's not a test, it's to raise awareness. So preclinical research uses a lot the MDX mouse. So I'm going to introduce it. It is a mouse that had a spontaneous mutation in its mouse dystrophin gene, so it doesn't produce dystrophin. And like patients without dystrophin, it has muscle pathology. But unlike humans, mice without dystrophin mainly have fibrosis, so not also the adipose, the fat infiltration that we see in patients. This mouse regenerates really well, and it makes big muscle. So it has hypertrophy, and that compensates for the loss of muscle strength. So if you look at this mouse in the lab, it does do quite well. But of course, in the lab, there's no predators. It gets all the food it needs to, say, feed the continuous regeneration. Survival in the lab is only slightly reduced. Probably in the wild, it would not live long because, well, the cat will get it. The pathology does get worse over time, but the trajectory is a lot slower than in humans. Now, if you were here last year, you will remember this question, but there's slightly different answers. So we'll just start easy. So we have the MDX mouse that I just introduced to you, and we're measuring the function of the diaphragm, so the muscle that we need for breathing. And you have in orange the regular function, it's 5, and now we've treated it with something in blue, and then the function goes from 5 to 8. So to get going, you scan the QR code, and then you get the questions. If the scanning doesn't work, you can also go to slido.com and enter 4108557. You also get to the questions. And then you can answer, and once you answer, we will see the results here. So, what do you think? Does this have potential? Yes, it improves the diaphragm, so it improves respiratory function. I'm slightly optimistic, but I want to see more about what's happening with the muscle, with the strength, or I do not have enough information to decide. We see people are answering. I'll give you some more time. And I think the majority is slightly optimistic, which it did do better. And then about 1 in 3, who probably were here last year, say, I want more information. Because there's one important thing missing, and that is, how well does a healthy mouse, a mouse with dystrophin, do? And here we see that one does 100, so the function here is 100. So if the healthy mouse had done 10, then going from 5 to 8 is quite an improvement. But we now know healthy is 100, and now this improvement, if you show it like this, it's a minor blip. So, well, we were optimistic, but seeing this, we're no longer optimistic. And this is really important. If you look at scientific research, see whether they also mention the healthy mice, the mice without the lack of dystrophin, because that will tell you how much of an impact did this make. So, we're going to develop another model. So, you're a preclinical researcher, and you've developed a frog model. There actually is a frog model. It's not yet published, but what I heard is it jumps less far and it swims slower. So there is muscle problems. This is my own invention. It produces excessive amounts of mucus on its skin. So you said, poor frog, I'm going to test a cream that reduces the mucus, and this works. So are you now going to test this in Duchenne patients in clinical trials? Yes, the model improves, because it should also work in Duchenne patients, or no, because I'm not aware that Duchenne patients have skin mucus problems. And we're laughing, and it sounds ridiculous, and I think the majority is saying, well, no, but sometimes a genetic model can have different symptoms. So this is AI generated, so I told Canva, like a frog with excessive mucus. I think it's more a frog that took a very foamy bath, but this is what AI came up with. So sometimes a genetic model will have symptoms that do not occur in the disease, in humans. And then some preclinical researchers will be focused, like these are the symptoms, these are the things that we're going to treat. And now this frog is, you will not forget this picture, this is why I put it in there. Of course, it's not always this clear. It might be very nuanced, where you think, well, this protein level that is elevated in the MDX mouse, now it's reduced back to normal. Yeah, but what does that mean for the patient? So this is really a message to you all. If people report, well, this is better, check, is this relevant for patients, yes or no? And if the drug developer can explain, yes, it's relevant because such, and that makes sense, fine. But very often, there's a tendency of preclinical researchers to develop a therapy for the model and not taking into account whether this is actually relevant for patients. So remember the mucus frog. So how do you then select your best model? We can have the MDX mouse, but there's certain questions we cannot ask of it, or we can ask for it. There's also a worm model, C. elegans, and this is an enlarged picture. The worm is really small in reality. And, well, worms, very small worms, easy to maintain. You can do studies in large numbers, but this worm does not have an immune system. Now, if we make a worm without dystrophin, it has muscle pathology. And it has functional effects. So if you see how quick is it crawling, it's crawling slower. And of course it moves like this, so the way that it crawls is different. Actually, what they also did, they sent the worms to the International Space Station, that's why the picture is there, to remind me to tell you this. So they sent the dystrophic worm and the healthy worm into space, and in space the pathology gets worse. Worse than what they called the ground control worms, which I thought was funny. Now, we're going to go back to our Earth location. So we have a lab here. We have the C. elegans worm that does not have an immune system. And now we have one that doesn't have dystrophin, and we're going to test corticosteroids. Do you think that this works? No, because steroids act by the immune system. This worm doesn't have an immune system, so it shouldn't work. Or, well, it might still work because maybe steroids do other things beyond targeting the immune system. I'll give you some time to think about this, and it's about 50-50. Which is interesting because what you would expect is this doesn't work because no immune system, steroids don't work. Steroids work in many different ways, and I'm going to look at Jared, who is nodding. They do many different things. We don't even understand completely all the things that they do in Duchenne. But if you give this worm without an immune system and without dystrophin, if you give it steroids, it improves. So definitely that shows it does more than just targeting the immune system. And had you asked me before this study was published, I would also have said, no, this is not going to work, but it does work. Now we're going to go into the mouse, back to the mouse. So we have our OG MDX mouse with decades of data on it. And I introduced it already to you. It's black because that's what its genetic background causes it to be. But we now have a new kid on the block. It has the same mutation, but it has a different genetic background. So you can see a picture. It's grayish, golden. But it has other differences as well because, well, it's different genetic background. And what we see, this model has more fibrosis at an early age. So with MDX mice, you have to wait really long because they get truly fibrotic. This one already at 3 months, a lot of fibrosis. Also, it has atrophy, so it has smaller muscle, where the MDX mice makes very big muscles. So there's this difference, but it's new. So what we don't know is, well, what about this mouse at 12 months? What about 18 months? How will it develop? So now we have a question. Imagine you're a preclinical researcher and you want to test a compound for 12 months. So we know for the D2 MDX mouse at 3 months it's more severe than a regular MDX mouse. But what you don't know is, well, if I start treatment at 1 month, at the end it will be 13 months. How does this develop? We don't know that. So what are we going to do? Are we going to do MDX mouse because we know what to expect, we know how to design? Are we going to test it in the D2 mouse and make some assumptions? Okay, Duchenne is progressive. It's not having dystrophin in a mouse, in the MDX mouse it's progressive. In patients it's progressive. I'm going to assume that in this D2 MDX mouse it's also progressive. Or you say, well, I have a lot of research funding. I look at Duchenne UK, they sponsor me, and I'm just going to do it in parallel in both models. And the majority seems to be saying, let's just do it in both. And I think that would have been an option, but definitely not do it immediately in the D2 MDX, because what we know now, I have to, of course, what we know now, the pathology at 3 months is more severe, and then somehow, after the mouse is about 5-6 months, it starts to recover. So the fibrosis goes away. How? We don't know, but we know that at the end point that you would have at 13 months, there's not that much difference between the wild type and the MDX, or the D2 MDX. So where you thought this is going to be a really good model, in reality, yes, if you do a short study, it would have been. But if you do a long study, it's not. So MDX would have been better. And then you can say, well, we just throw a lot of money at it and we do it in parallel in both. But just imagine the waste, like fundraising takes a lot of time and effort. If you make the study twice as big, it will be twice as expensive, and you're now wasting, well, a lot of money that you could have used to study something else, on top of wasting a lot of mice that now give their life for not really any information. There's other problems that we now realize because Duchenne UK sponsored a big national history study for this model. We know it has calcifications in the muscles and in the heart. And this happens even in the healthy mouse. So the mouse with dystrophin starts having calcium deposits in its heart. So it starts to have heart problems. Now, if we're testing something that should improve heart pathology, and we're seeing there's improvement, what we don't know is it targeting the pathology that arises from lack of dystrophin, Or is it targeting the pathology that is just occurring in this model that's not really relevant for Duchenne? So are we targeting the disease or are we targeting the model? So you have to really wait and see which model do you use when. And also here again, when you read the scientific report, just see, well, does this make sense or not? And did this research really answer the questions that they should have asked? So briefly something about mutation-specific treatments. I know after the break there will be talks about exon skipping. You might have heard about genome editing. These are approaches that are mutation-specific. So they're also specific for human sequences and for mouse sequences. So all the exon skipping that we've done in the MDX mice, which works really well, applies to 0 humans. And all the things that we can develop for humans do not work in the mouse. So that is the first problem. So when we go from the mouse to the human, we use a different treatment. The second problem is that the location of the mutation is different. So the MDX has a mutation exon 23. Most Duchenne patients have a mutation somewhere between exon 40 and 55. So the dystrophin that will be produced after you do your exon skipping in the MDX is probably more functional than the one that we have in patients. They're both partly functional, but partly is a flexible term, and some can be more functional than others. So now when we go from mouse to human, we use a different species. Mice are not human, human are not mice. We use a different therapy because, well, the human one doesn't work in the mouse and vice versa. And it results in a different dystrophin. So that makes extrapolation very difficult. So what we've developed at LUMC is humanized mouse models where you can test your human-specific treatment, gene editing or exon skipping in the mouse model and then develop a dystrophin that you would make in humans. So they're available at LUMC, but we share. So we've shared them already with many academic collaborators and licensed them to companies. So briefly something about supplements. And I'm sure you've all heard of supplements. This is just one example, but there's many. You can get food supplements. You can get them over the counter. And this is one, so I have no stock in this whatsoever. This is just one that was a publication about, so I use that as an example. It's beta-glucan. You can see the chemical picture there. A random one, there's different brands. I just pick one from the internet. And what this has, it has anti-inflammatory and anti-fibrotic properties. Well, we know Duchenne patients have inflammation and they have fibrosis. So, knowing this, if you're a Duchenne patient, would you take this? If you're a parent, would you give this to your child? Yes, because it might do something and it's over the counter, so it's safe. I will wait for clinical trial results. I want to see what's happening in a model system, or I would never use this. And again, there's no right or wrong. I'm asking what you would do. And well, most people want to wait for the clinical trials, which is, I think, good. But there's also some that say, well, it's available. About 1 in 5 says it's available over the counter, so what harm can it do? Well, bear in mind, you might think supplements are safe because you can get them without a prescription, but that is not really the case. So if you take more than the recommended dose, it might give toxicity. And that's not because of the supplement, but because there can be impurity in there. It's not a drug, it's a supplement. There's less restrictions. So don't overdose yourself. Don't think more is better, because more might actually be toxic. Also, it's less controlled. So the dose, like you're not, like if you get 30 milligram givinostat, you get 30.0 milligram givinostat. Here, well, it might be anywhere between 25 and 35. It's not that well regulated. Be aware that supplements can have negative interactions with drugs you're taking. So there can be side effects that happen just because you're taking steroids or ACE inhibitors and a supplement, or the supplements can now mean that your steroids are not working as they should. So be aware of that. The fact that it's safe in a regular, healthy individual doesn't mean it will be safe in Duchenne patients, because there's a lot of muscle breakdown and the kidneys have a problem with that. So that they cope, but if you're now adding something else on top of it, it might be a problem. So if you consider taking these supplements, discuss this with your clinician. And if your clinician says, well, it's not going to work, that is not the question that you're asking. The question that you want to know is, will this have a negative effect on the other treatments that my son is taking? So discuss this. Back to our paper. Now, what they did, they had mice, MDX mice, 3 weeks old. They had wild-type controls, hooray, and they were treating the mice with beta-glucan for 45 days. And there was no effect whatsoever on muscle strength, muscle function, what the muscle looked like. But in the text, the authors say, and also in the title of the paper, the dystrophin levels are increased. Well, first of all, why should the dystrophin levels be increased? Like, this does nothing to the gene. Why should this MDX mouse suddenly be able to make dystrophin? Doesn't make sense. Now, also, if you look at the figure, so they claim in the text and the title, dystrophin is increased. This is what they're referring to. Well, so this is MDX untreated, this is MDX treated. This apparently is more than this. This you may say, well, might slightly more than that, but this NS means not significant. So the claims that they make don't fit with the data that they show. And this is another thing, and it might be difficult to interpret all these graphs, but just be aware that what people say might not always reflect the data that they show. So you need to look into the data. Then, slight move, you can of course also repurpose treatments. And that means you have a drug that is approved for one disease, and maybe it will also work for Duchenne. And development can be quicker because trials were done, it's registered, we have human data. But there's consideration. So consideration first, if it works in the MDX mouse, test the dose. Sometimes they overdose the mice, and then you see, well, but this is never ever a dose that we could use in humans. Also, is the drug approved for an acute disease or for a chronic disease? So if it has severe side effects and it's a lethal disease, you might be willing to take that drug for a week and then not die and then stop the treatment. For Duchenne, you will have to take it the rest of your life. Is that tolerable? Is there data available for children? It might be approved for the elderly, and then there's no data for children, so it's not that quick. And again, safety in one disease doesn't mean it will also be safe in Duchenne, because there might be interactions with the steroids, with the ACE inhibitors, and there might be other side effects, as I mentioned for the supplements. I'm going to end with a last case, and this is actually a collaboration that we did with Duchenne UK. So a while ago, there was a publication that simvastatin, which is a statin, so like against high cholesterol, but it also has anti-inflammatory and anti-fibrotic properties. So a group in the US said, we are going to test this in the MDX mouse, and what they found was, in the MDX mouse, this improves muscle pathology. So there's less fibrosis, and it also increases diaphragm function. And they had their wild-type mice. It wasn't as my first example. They saw a significant improvement. So they said, we now want to test this in clinical trial. And they went to Duchenne UK. They said, can you fund our clinical trials? But Duchenne UK had also been involved with TACT, so they knew we need independent validation. So what they asked us at LUMC, can you repeat this study and see whether it really works? Because, well, we just want to know how robust this is. So we tried to repeat it. We tried to do exactly what the group in the US did, and we were unable to find an effect. So we saw no effect on fibrosis and no effect on diaphragm function. So what we thought was, okay, we did something wrong. But then Duchenne UK was clever, so they also asked another group in London to do the same experiment, and they also could not find it. So also they found no effect on fibrosis, no effect on function. So then we could team up, and together with Duchenne UK, we had this publication, like, okay, there is this publication out there that it might work, but what we see now is that actually, in our hands, it doesn't work. And this brings me to the last problem, a problem of replication. So I'm not saying that the first publication was false and that they were fraudulent in any way. They found something, but it was not possible for us to replicate it. And actually, there's a lot of research that cannot be replicated, even by the people in their own lab. So it means that the treatment, whatever effect, it might have just been chance, or there might be a minor treatment effect, but it's not robust. Well, that if it's not robust in mice, it's never ever going to be robust in humans, because there's a lot more variability between individual humans living around the world than mice that live in a laboratory cage. So it's really important that there's independent replication done. It's difficult to publish that you were unable to find the same thing that was previously found. We were lucky because there were two groups doing the same thing, so we could team up. That made our case stronger. But now there's also bioRxiv, and that's just a big, well, catalog or collection pot where people can submit research, where they say, well, it's important, we did everything, we can't get it formally published, but we think it's important that the world knows this. So also there, check if there's this new weird trial compound that you never heard about, just check. Maybe there's something on bioRxiv where it says, well, we tried this, but it didn't work. So, take-home messages, model systems have limitations. There are no perfect models. The perfect model is the human. But we can, of course, use the models to test our research questions. So when you hear anything, use your common sense and be critical. Did they use controls? Did they use the healthy control? Did they do replication studies? Are the symptoms that they are treating, are they relevant? They say one thing, does it match the data, etc. And finally, if it sounds too good to be true, it probably is too good to be true. So I think that is your first thing. If it's too good to be, if it was easy to solve Duchenne, like, we would have done it already. So that is the last question. And with that, I thank you very much for your attention and happy to answer questions if there are any.